Hi friends, you are back with me, Professor Girish Kukreja. Today, what we'll be talking about is the photosynthetic carbohydrate synthesis. Yes, today what we'll be talking about is the Calvin cycle. Most of the time students are confused with the Krebs cycle and the Calvin cycle. Please don't get confused. One is the oxidative one and this one is the reductive one. There we are talking about the Krebs cycle, the TCA cycle, that is in the respiration. This Calvin cycle is, as you know, the second phase in the photosynthesis. If you remember in our earlier videos, we have talked about the light reactions, the light dependent reactions, the photophosphorylations, where by using the light energy, we had synthesized ATP and your NADPH, right? So using this ATP and NADPH will be what you call as not we don't do this, you know that it is the work of your green plants, your cyanobacteria, your algae, which are actually going to reduce your carbon dioxide to your glucose. Yes, and this particular what you call as reduction or fixation of carbon dioxide as you call is occurring via a cyclical process where the intermediates are regenerated again and again. That particular cycle is called as the Calvin cycle. Yes, so you got a brief summary. So Calvin cycle, we know that is the second phase after the photophosphorylation, after your light dependent reactions. These cycle also sometimes popularly called as the dark reactions, but then this leads to what you call as conception that this particular Calvin cycle is not dependent on the light reactions. It's not like that. Actually, there is no direct involvement of light in the Calvin cycle, but yes, it uses the products of the light reaction. The two major products of the light reaction which the Calvin cycle would be using is your ATP and the NADPH, the energy and the reducing power, right? So the energy in the form of ATP and the reducing power in the form of NADPH will be using in this particular Calvin cycle. This entire pathway, the cyclical pathway of carbon dioxide fixation taking place in these photosynthetic organisms was worked out in around 1950s by Dr. Melvin Calvin, Andrew Benson and James Bashman, and therefore this particular cycle is called as the Calvin cycle. When the experiments were being performed by Dr. Melvin Calvin, he illuminated the green alga with 14 CO2, right? C14 CO2 we can say, and later on, immediately after few seconds, these alga they were killed. And then by chromatographic methods, they were trying to find out where did this carbon it localized itself. And it was found that the first stable product which could be identified immediately after illumination and killing the cells and chromatographic separation of the molecules was a 3-carbon compound, that is your 3-phosphoglycerate. And therefore, this particular pathway also called as the C3 pathway. C3 pathway, a common pathway for formation of carbohydrates in all photosynthesizing organisms. You must have heard about variations in these also. You must have heard of organisms like when I talked about C3, probably you must have recalled there is something called as C4, there is something called as CAM. So whether it is C3, whether it is C4, whether it is CAM, remember this pathway operates in all of them. So C3 pathway operates in C3, no doubt. It also operates in C4, it also operates in the CAM plants, remember. So now let us come to this particular pathway, that is your Calvin cycle. So basically talking about this Calvin cycle, as I told, comprises of 3 cyclical steps. Stage 1 is what is called as the carbon dioxide fixation. So carbon dioxide fixation, or you can say the first phase, that is carboxylation. So in this particular first stage, you will find that there is a 5-carbon acceptor RUBP, ribulose 1,5 bisphosphate. So a ribulose 1,5 bisphosphate, it combines with what is called as your carbon dioxide, and there is formation of a 3-carbon intermediate, that is your 3-phosphoglycerate, Remember, so there is formation of what is called as your 3-phosphoglycerate. This particular reaction is catalyzed by the world famous enzyme that is your Rubisco. Mercifully, what you call as shortened the name, it is actually ribulose 1,5-bisphosphate carboxylase oxygenase. I repeat for all of you, it is ribulose 1,5-bisphosphate carboxylase oxygenase. Now this is the enzyme which we popularly refer to as Rubisco, which is catalyzing the first step of carboxylation. So in the first step, your 5-carbon acceptor RUBP, ribulose 1,5-bisphosphate, accepts carbon dioxide. There is a formation of a 6-carbon unstable intermediate which is immediately cleaved by the Rubisco itself to form 2 molecules of your 3-phosphoglycerate, right? So 1 RUBP, 1 carbon dioxide, and you have 2 molecules of 3-carbon 3-phosphoglycerate, right? So this is the first step catalyzed by Rubisco. Rubisco, as I told, like feeding enzyme which we may call, which is fixing the carbon dioxide for the entire biosphere, and we all are surviving through that. So this particular feeding enzyme, you will find that in plants this enzyme works comparatively slowly. You will find the turnover number of this enzyme is hardly, it fixes 3 molecules of your carbon dioxide per second per molecule of this Rubisco at 25 degrees Celsius. In a typical actively photosynthesizing chloroplast, you will find that around 50% of the protein, it is this particular what you call as your Rubisco. Rubisco is one of the most abundant enzymes in the biosphere. There are many more interesting facts related to Rubisco, the way it is regulated. Stay tuned with us to know more about this particular Rubisco. So talking about this particular Calvin cycle, talking about the first stage. So in the first stage, your RUBP 5-carbon intermediate combined with carbon dioxide. So 5 plus 1, 6, and 2 molecules of your 3-carbon intermediate, that is 3-phosphoglycerate. The second step is what is called as your reduction. So in the second step of reduction, which again occurs in 2 stages. First, there is phosphorylation, where your ATP is converted to your ADP, catalyzed by what is called as your 3-phosphoglycerate kinase, which will add a phosphate group, and it will form an intermediate of 1,3-bisphosphoglycerate. This is then reduced by glyceraldehyde 3-phosphate dehydrogenase by using the NADPH which we have formed in the light reaction. So these are all the products of the light reactions. So your reduction takes place, and there is formation of this glyceraldehyde 3-phosphate. Now this glyceraldehyde 3-phosphate, it can isomerize with DHAP, the dihydroxy acetone phosphate. Your triose phosphate isomerase enzyme can carry out this particular isomerization. So we have come to the second stage where there is formation of glyceraldehyde 3-phosphate. Say for example, if you are looking for the formation of a triose phosphate, you started with 3 molecules of RUBP, 3 molecules of RUBP, 3 molecules of carbon dioxide, so you will have 6 molecules of your 3-phosphoglycerate. Out of these 6 molecules, 1 molecule would be used for what you call as synthetic purposes. So here you will find this particular 1 molecule here, it can go for working as a fuel, right? It may immediately undergo glycolysis. It may what you call as go for synthesis of starch. So your glyceraldehyde 3-phosphate, DHAP, they can combine together to form fructose 1,6-biphosphate to fructose 6-phosphate, which can be converted to starch. It can be converted to sucrose, where it can be later on transported to the non-photosynthesizing parts of the plant. So it may be converted into that. So this is the final product, your glyceraldehyde 3-phosphate, which you can use for multiple purposes. U means the green, what you call as plants. I am talking about the algae, the cyanobacteria. So this particular molecule, 1 molecule is the net product. So whatever 3 carbons have entered here, they are now what you call as can be used as fuel. They can be used for formation of starch. They can be used for formation of sucrose to be transported, and for n number of what you call as purposes. Out of this now, out of the 6 molecules of glyceraldehyde 3-phosphate you had, 1 is your net product. You have 5 molecules. Now these 5 molecules of your glyceraldehyde 3-phosphate isomerizing with DHAP can be used in the third stage of what is called as your regeneration of the acceptor. So your 5 molecules of glyceraldehyde 3-phosphate are rearranged with the help of different enzymes like aldolases, transketolases, transaldolases and phosphatases to form what is called as your 3 molecules of your RUBP, that is ribulose 1,5 bisphosphate. So you have again came here. So 3 cyclical steps in the Calvin cycle: the fixation, the reduction and the regeneration, and 1 molecule it is for the synthesis. So here you had regeneration of using 5 molecules, and here you had your 3 molecules which are reformed. So this is how it takes place. The Calvin cycle, it takes place in the stroma of the chloroplast. So in eukaryotic organisms, we know that the light reactions, they take place in the grana and thylakoid membranes, whereas the dark reaction, it takes place in the stroma, which is bounded by the inner membrane. So all the enzymes of what you call as your Calvin cycle, they are actually present in the stroma. If you talk about the enzymes which are involved in the entire process of Calvin cycle, you will find most of the enzymes are also present in animal tissues. We lack 3 enzymes possibly. One is the major enzyme, that is Rubisco. Second is your pseudoheptalose 1,7 bisphosphatase. And third is your ribulose 5-phosphate kinase. So these are actually the enzymes which we lack. Otherwise probably we would have also performed photosynthesis. So it would have been like, if you want to have a lunch, you will go in the sun for half an hour. If you want to have a breakfast, you will go in the sun for 15 minutes. But it is due to the lack of these 3 basic enzymes that we actually cannot have a net conversion of this carbon dioxide into glucose into us. Anyways, so summarizing this Calvin cycle, if you talk about the stoichiometry of this particular Calvin cycle, you have, say if you start with 3 molecules of your carbon dioxide entering, combining the 3 molecules of your ribulose 1,5 bisphosphate in the first stage, that is your carbon dioxide fixation. So you have 3, each one of these is 5 carbons, so you have a total of 15 carbons. 3 carbons have entered in, so it is 15 plus 3, how many? 18. So there is fixation. Remember the enzyme is Rubisco. Then this particular Rubisco, 15 and 3, 18, these each is of 3 carbon atoms, 1 RUBP, 1 CO2, 2 molecules of 3-phosphoglycerate. I have 3 RUBP, 3 CO2, so I will have 6 molecules of 3-phosphoglycerate. These are phosphorylated, as I told, by 3-phosphoglycerate kinase. So you require 6 molecules of your ATP getting converted to 6 molecules of your ADP to form 6 molecules of 1,3-bisphosphoglycerate. So 18 carbons still there. Then we have reduction by glyceraldehyde 3-phosphate dehydrogenase. You have NADPH and H plus getting what you call as oxidized here. 1,3-bisphosphoglycerate getting reduced, and yes, there is a removal of phosphate here, and 1,3-bisphosphoglycerate, 6 phosphate molecules would be removed, and 6 molecules, yes, of NADPH will require to form 6 molecules of glyceraldehyde 3-phosphate isomerizing with your DHAP. Out of these 6 molecules, 1 molecule is your net product. This 1 glyceraldehyde 3-phosphate you can use for synthesis of sucrose, which may be used for transportation, for synthesis of starch, which may be stored. You know that this is taking place in the chloroplast stroma. When you have a lot of starch to be stored, it is stored in the amyloplast, which is also a plastid. Or this particular can be used as a fuel directly. This can enter glycolysis and it can be what you call as used as a source of energy. So you have 1 molecule as a net product, the 3 carbons. So whatever 3 carbons you had here fixed, they are now in the form of this glyceraldehyde 3-phosphate. So there is net this formation. We will be going for the next step of regeneration. For regeneration, how many molecules will be using? 5. So 5 molecules of glyceraldehyde 3-phosphate, each of 3 carbons. So again we have 15. With 2 molecules of phosphate, doing all types of rearrangements with the intermediate formation of 4 carbon sugars like erythrose 4-phosphate, 7 carbon like pseudoheptalose 7-phosphate, and many more. You will find there is rearrangement of these to form 3 molecules of 5 carbon ribulose 5-phosphate. So if you look at the number of carbon atoms, they are balanced. It's 5 into 3, or 3 into 5? It is 15. We started with 15. So ribulose 5-phosphate, phosphorylated by ribulose 5-phosphate kinase. So you need ATP. So 3 molecules of your ATP phosphorylated to form 3 molecules of ribulose 1,5-bisphosphate. So you have finally regenerated. So this ends your Calvin cycle. So if you look here, to form 1 molecule of triose phosphate, we required, this is 6 molecules of your NADPH, and we required these 6 plus 3 molecules of your ATP, that is 9 molecules. So for net fixation of 3 carbon dioxides to form 1 molecule of glyceraldehyde 3-phosphated triose phosphate, we require 6 molecules of your NADPH and 9 molecules of your ATP. So ratio of 2:3. If you look at this, this is the similar ratio in which you form these in the light reaction. If you want to synthesize 1 glucose molecule out of this Calvin cycle, now these were the 3 turns of Calvin cycle. If I want to form a molecule of glucose, which will require 6 turns of the Calvin cycle, tell me in the comment section, for formation of 1 molecule of glucose, you will require how many molecules of NADPH and how many molecules of your ATP. I hope you were with me in this entire process of Calvin cycle, and thank you so much, and do tell me how many NADPH and how many ATP for 1 molecule of your glucose. Stay tuned with us. Thank you.